Hello everyone, welcome to my YouTube channel. In this video, we will talk about the cell. When we talk about the cell, the very first question that comes in our mind is that what is a cell and why do the organism have cells? Cell is the basic fundamental structural and functional unit of all living organisms. Organisms have cells because all the important functions, metabolic reactions occur in cell. So there are two types of organism. First is unicellular organism and another is multicellular organism. Unicellular organism are those organisms which is composed of single cell, and multicellular organisms are those organisms which is composed of numerous cell. So unicellular organism have shorter lifespan and they carries all the life functions in a single cell, and they have great regeneration capacity, for example amoeba, euglena. And in multicellular organism, different cells are specific for different functions. They have longer lifespan and they have poor regeneration capacity, for example animals and plants. Now, cells are enclosed by a thin film layer membrane called plasma membrane, also known as plasma lemma or cell membrane. They form a body boundary around the cell and does not allow any unnecessary material to enter into the cell. That means the cell membrane is selective in nature, that allows only selective material to enter into the cell. Now let's learn about the structure of the plasma membrane. So there are so many models given by different scientists, but the most accepted one is fluid mosaic model, which is given by Singer and Nicholson in 1972. And this model states that the quasi-fluid nature of lipid enables lateral movement of protein within the overall bilayer, and the ability to move within the membrane is measured as its fluidity. Lipid bilayer are amphipathic in nature, means they are both polar, that is hydrophilic, means water loving, and non-polar, that is hydrophobic, that is water repellent. Now polar side faces the extracellular matrix that is present outside the cell, that is known as extracellular matrix, and the non-polar side faces each other. Now the peripheral protein are those protein that is present on the polar side, and the integral protein are those protein that are embedded on the membrane and present on both polar and non-polar side. As you can see in the picture, membrane protein that is present at the periphery of the membrane, and there is a integral membrane protein that is embedded into the membrane. You can see some cholesterol also and some glycoproteins and glycolipids also. There is present some channel proteins also. Now these proteins act as a gatekeeper, act as a channel, as I said, which allows only selective molecule to enter inside the cytoplasm of cell, method by which the transport occur across the cell membrane. First is active transport. In this transport, the ATP or energy is required because it occurs against the concentration gradient. Against means the molecules are moving from low concentration to high concentration. And the another transport is passive transport. In this transport, no ATP or energy is required because the transport occurs according to the concentration gradient. According means the molecules are flowing from higher concentration to the lower concentration. In passive transport, there can be simple diffusion or passive type of diffusion. Now, what is a cytosol? So cytosol is a fluid-like structure that is present inside the cell membrane and surrounds the organelles present inside the cell, like mitochondria, Golgi apparatus, endoplasmic reticulum, lysosomes, etc. Cytosol is composed of 70 to 80% of water and of various other ions like glucose, amino acid, ATP, protein, lipid, etc. Now, organelles that is present inside the cell performs specific functions in cell growth. They perform specific functions in cell maintenance and reproduction, and divided into two types: non-membranous and membranous. Non-membranous are those organelles which do not have any membrane, like cytoskeleton, cilia, flagella, etc. And membranous structures are those who have membrane, like mitochondria, endoplasmic reticulum, lysosome, Golgi apparatus, etc. Now let's talk about the mitochondria. So mitochondria is a double membrane bound structure, and it is divided into two compartments: outer compartment and inner compartment. The inner compartment is filled with dense homogeneous substance which is known as matrix, and the inner compartment has several foldings which is known as cristae. Mitochondria is also known as the powerhouse of cell because it produces lots and lots of energy. It is present in the aerobic organisms and possesses circular DNA, few RNAs, etc. It is divided by the fission. As you can see in the diagram, there are the inner membrane and the outer membrane, and in between the membranes there is intermembrane space, and in the inner membrane there you can see the matrix and cristae, and in the matrix you can see some granular structure, that is ribosome, and some ATPase also that helps in the synthesis of ATP, and also you can see some mitochondrial DNA. Now let's talk about the endoplasmic reticulum. Endoplasmic reticulum provides the skeletal framework to cell, means it supports the structural framework, or it gives the structure to the cell, and it is attached to the membrane of nucleus. It helps in the exchange of molecules by the osmosis and diffusion. The important function of endoplasmic reticulum is protein synthesis, and they synthesize and store lipid. RER and SER. SER help in the synthesis and metabolism of lipid and steroid, and RER helps in the protein synthesis and secretion. RER are continuous with the outer membrane of nucleus. As you can see in the diagram, RER is continuous with the outer membrane of nucleus. It is attached to the nucleus, to the outer membrane of the nucleus, and contains some granular structure. That red dotted structure is the granular structure which is present in the RER. That's why it is called RER. And the portion where this granular structure is not present, that is known as smooth endoplasmic reticulum. Now let's talk about the Golgi apparatus. It is composed of many disc-like sac or cisternae, that sac is also known as cisternae, which are stacked parallel to each other, and it has two phases: cis phase, that is known as forming phase, and trans phase, that is known as mature phase. Golgi apparatus helps in the packaging of material and deliver them in the cell or out of the cell. Golgi apparatus act as a temporary storage place for protein and other component that is synthesized in the ER. Acrosome present in spermatozoa is derived from Golgi apparatus, and Golgi apparatus also help in the synthesis of carbohydrate components of plasma membrane. Now we all know that Golgi always remain in association with ER because the material that is synthesized in the ER is transported to Golgi, and material first packed into the vesicle from the ER. As you can see in the picture, the material is packed into the vesicle from ER, and that vesicle fuses with the cis phase. As I said that Golgi has two phases, cis and trans, so that vesicle fuses with the cis phase of the Golgi and then moves towards the trans phase. Now let's talk about the ribosome. As I said, that ribosome is a granular structure that is composed of RNA and protein. Eukaryotes have 80S ribosome and prokaryotes have 70S ribosome. Each ribosome have two subunits, larger subunit and smaller subunit. In eukaryotic 80S ribosome, the two subunit is 60S and 40S. And in prokaryotic 70S ribosome, the two subunit is 50S and 30S. The main function of ribosome is the synthesis of protein. That's why the ribosome is known as the protein factory of the cell. As you can see in the picture, the ribosome have two subunits. The smaller subunit, which is the blue one, is the smaller subunit, and the maroon one is the larger subunit. And the average length or size of the ribosome is around 20 nanometer. Now let's have a look on lysosome. So lysosome is found in the cytoplasm and formed by the process of packaging of Golgi. Lysosome is known as the suicidal bag. Why it is known as suicidal bag? Because it contains the hydrolytic enzyme like lipases, proteases that helps in the digestion of proteins, lipids, carbohydrates, nucleic acids, etc. The inner environment of the lysosome is slightly acidic and the pH is around 4.5. Lysosome is covered by the single membrane. Now, cytoskeletal structure. It is an intracellular framework which is composed of microtubules, microfilament and intermediary filament. It supports the structural framework, or it gives the structure or shape to the cell and involves in the cell movement. It is important in the cell division also. As you can see in the picture, the intermediate filament, microfilaments that is present all over the cell, and these filaments gives the or provides the structural support to the cell. Now let's talk about the cilia and flagella. Cilia and flagella are the hair-like structure and they form by the extension of the cell surface or cell membrane and helps in the cell movement. Both cilia and flagella are the extension of cell membrane, as I said. They contain the cytoplasmic matrix and with 9+2 arrangement of microtubule. Flagella of bacteria, they do not show the 9+2 arrangement. Now let's have a look on their difference. Cilia are numerous and short. Cilia have an average length and they beat together, but in case of flagella they are few in number and long. Flagella can be of 150 micrometer to 510 micrometer and they beat independently. Now let's talk about the most important component of the cell, that is nucleus. It is also known as karyon and its study is known as karyology. Nucleus is a primary carrier of hereditary material in cell. It is divided into 4 parts: nuclear membrane, nucleoplasm, chromatin and nucleolus. Now let's talk about the nuclear membrane. The membrane that surrounds the nucleus, or the nucleus which is enclosed into a membrane, that membrane is known as nuclear membrane. Nuclear membrane is double layer and porous. Why porous? So that the material that is formed inside the nucleus or the material that is formed outside the nucleus can cross the membrane, so that the membrane of the nucleus is porous. And between the two nuclear membrane there is a space which is known as perinuclear space. The outer layer is connected to the endoplasmic reticulum and may contain RER and the inner layer contain SER. During the time of mitosis and meiosis, it always gets disappeared. Now, nucleoplasm. Nucleoplasm is a fluid that is present inside the nucleus. The fluid is also known as nuclear sap and contains DNA and RNA polymerase that helps in the synthesis of DNA and RNA. As you can see in the picture, There is a nucleus, and inside the nucleus a fluidy material is known as nucleoplasm, and inside the nucleoplasm you can see a thread-like structure which is known as chromatin. Now let's learn about the chromatin. Chromatin is present inside the nucleoplasm. Chromatin is converted into the chromosome at the time of cell division. It is present in nucleoplasm and contain the hereditary material. Means inside the nucleus, nucleoplasm is present, and inside the nucleoplasm chromatin is present, which contains the hereditary material like DNA, protein, etc. The protein which is present inside the chromatin is histone protein, and that circles the DNA. So there are around 4 types of histone protein and 2 of each: H1, H2A, H2B, H3 and H4. Now nucleolus, it is present inside the nucleus and attached to NOR, that is nucleolus organizer region. It consists of DNA, RNA, protein, but largely composed of RNA.